Okay, welcome back for chapter 4. We have one more chapter, then it will be time for exam 1. I very strongly recommend after the conclusion of this chapter that you go back through the summary lectures that we'll be going over in class and use that as kind of a study guide to help you out. That's going to be the big ticket items. You will still need to review the vocabulary, and I recommend at some point going through the PowerPoints at least one more time before the exam. But if you understand all the material on the summary lectures, then you should be in pretty good shape for the exam. So let's crack straight into chapter 4 here, get this out of the way, and then I will see you in class. So membrane structure and function. So the membrane structure and function regulates movement of molecules in and out of the cell. Phospholipid bilayer embedded with proteins. You have this hydrophilic, water-loving polar heads, hydrophobic, water-fearing, nonpolar tails. And then in animal cells, you also have some cholesterol in the cell wall. The plasma membrane structures and functions. So you have membrane proteins that may be associated with the peripheral proteins, meaning they're on one side. They often have structural roles and help stabilizing the shape of other proteins. Integral proteins span the length of span through both sides of the plasma membrane. Let's see if we can zoom in here a little bit. So channel proteins just allow things to move through, usually with the concentration gradient. Carrier proteins allow specific things to move through. Cell recognition proteins, this is your major histone compatibility complex. This is what tells your body that you are you, and interestingly enough has a lot to do with how you smell, among other things. And the more closely related someone is to you, the worse they smell to you. It's thought to be a very simplistic way of making sure you don't inadvertently reproduce with anyone who's too closely related, because you don't like the way they smell. You have enzymatic proteins that catalyze reactions, and then you also have receptor proteins. So typically a hormone or something will dock on here, and that will send a signal through to the inside of the cell to regulate some type of function. Permeability of the plasma membrane. So it is selectively permeable, meaning some things can go in and some things can't. And this is influenced by the size, the nature of the molecule, meaning like polarity and charge, concentration gradients. Typically things will move down a concentration gradient from from high concentration to low concentration. If you want to go up a concentration gradient, that requires the input of energy, and you'll have to use a pump to do that. Some molecules can move freely across, such as water, small non-charged molecules. Sometimes you have little things called aquaporins to allow water to cross the membrane more freely. Other molecules that cannot pass through are water soluble, such as sugar. You actually have to have a specific gate to allow sugar into the cell, and that's why you have that's why your body produces insulin, because the glucose transporters on the majority of your cells are insulin dependent. So you cannot take up the glucose from your blood without insulin, except in nervous tissue, because your nervous system can only function on two things, ketone bodies and glucose. And it takes about a week or so for enough ketone bodies to build up in your system to be able to fuel the brain. So it's always pouring in glucose into the nervous system, which is why if your blood sugar goes too high, you start damaging the nervous tissue, because it causes an influx of water following all that sugar into the nervous tissue and can cause the cells to lyse. And we'll talk a little bit more about how that functions in a minute. Passage of molecules in and out of the cell. So you have diffusion, facilitated transport, active transport, exocytosis, and endocytosis. Basically, just look over the chart here and Don't worry about memorizing it. Try to think through it and understand the concept, and you'll be good. So permeability of the plasma membrane. So if you're going to diffuse, you move down a concentration gradient. It does not require energy. Gases tend to diffuse through membranes, like in our lungs. So this is an alveoli here with these little capillary beds around it. You are diffusing oxygen into the blood, because by the time it comes back around to the lungs, it's oxygen depleted, and then you are diffusing CO2 out of the blood and into the gaseous space in the alveoli, because at this point it's full of CO2. Which, incidentally, that's why you weigh less in the mornings, because all night you're breathing in CO2, or breathing in oxygen and breathing out CO2, meaning that for every molecule exchange there's an extra carbon leaving, which is heavier. So you literally breathe off mass constantly. And we'll get into that a little more later. Several factors influence the rate of diffusion, such as temperature, pressure, electrical currents, and even the size of the molecules and the difference between the concentration gradients. So water has a major impact on cell activities and survival. Osmosis is the diffusion of water, and it always occurs from higher to lower concentrations. Osmotic pressure is a pressure that develops in systems due to osmosis, and this osmotic pressure is what can rupture cells, like we were talking about before. And this happens when you have a selectively permeable membrane and the relative concentrations on either sides of it are out of balance, so meaning one side's more concentrated than the other, and these solutes cannot pass through the membrane, but the water can. So the water will diffuse through the membrane and will create equal concentrations on both sides, which is what's happened here in this little experiment. And the level in the beaker here, or in the beaker here, has risen. Got the hiccups today. Okay, terms of tonicity. You have hypotonic. Hypo meaning less or under, tonic, solute concentration, so less solutes, thus more water. And then you have hypertonic, which is exactly the opposite, more solute, less water. And then isotonic, everything is equal on both sides. If you place a cell in an isotonic solution, it's going to be happy, no net movement of water in or out, everything's cool. The hypotonic solution, the cell will gain water, it'll swell up and burst like a balloon, and then in a hypertonic environment, the cell will lose water and crenate. It basically shrivels up like a grape, which can also kill the cell. Plant cells are a little different. The main thing is when you place them into a hypotonic environment, the vacuole in the middle will swell, and you will see some bulging in the cell wall, but the cell wall provides added rigidity, so it kind of stays together, and plants actually use this to create turgor pressure to keep them upright. So they're kind of like pressurized water balloons, and it's the membrane of, or it's the cell wall is the membrane of the balloon, kind of keeping everything together. And then if you place them in a hypertonic environment, they will shrivel up inside the cell wall, but the cell wall is still there to provide some support, but you can still easily kill the cell with this plasmolysis. It just dies, but it still maintains its shape, which is useful. Permeability of the plasma membrane. So we have carrier proteins, and they're required for facilitated transport and active transport. And carrier protein is just anything that combines with a molecular ion to transport it across a membrane. Some of them, like for facilitated diffusion, they just let things pass through. It's basically just like a gateway that they can move through. It doesn't really require energy. But for active transport, you have to burn energy in order to pump something against the concentration gradient. And the classic example of that is the sodium-potassium pump. For larger things, you can form a vesicle and then merge that vesicle with the plasma membrane to excrete things through via exocytosis. So you've got this vesicle here, and it's this nice little double membrane here, and it merges with this one and empties the contents. For endocytosis, you kind of form this invagination here, which closes itself off and then forms a vesicle, and it's basically just this in reverse. And that's it for chapter 4, and I will see you in class.